Okay, so today we're going to be talking about the immune system again. This is part 2 of our series on the immune system. This part is on adaptive immunity. So this goes back to a previous slide that I showed you before. Last time we talked about innate immunity. As I mentioned, this is a pretty fast kind of immunity that you're going to have, something that you're born with. So we're talking about on the order of minutes. And we broke this into two parts. The first part was physical barriers, mostly including the skin and skin secretions, also including your normal bacterial flora. The second line of defense were chemical and cellular responses, so including various types of cells and processes such as inflammation involving chemicals. Today we're going to be talking about adaptive immunity. So adaptive immunity involves lymphocytes, these are B cells and T cells, and antibodies and a whole host of other things. This is a specific memory, meaning it remembers something specifically that your immune system or your body has been exposed to before. So, talking about adaptive immunity, let's talk about some of these words we're going to use. First is specificity. So what does that mean? That means that the immune system can recognize a specific molecule or something on the cell surface of a cell or something on the surface of a virus. Inducibility. Inducibility means that the immune system is turned on when it's needed and not turned on when it's not needed. Clonality. So this has a lot to do with these immune cells. These cells can clone themselves and make duplicates of themselves. This is important for both immune action and also important for immune memory. Number 4, no response to self. This is very important. We do not want our immune system to attack its own body. This would be an autoimmune disease. This is not good. So we want to make sure that doesn't happen. And lastly, memory. So as I mentioned before, we want to be able to remember what we saw before, what made us sick, and then the second time around have a secondary immune reaction that will hopefully not allow us to be sick at all. So all of these words will come up as we talk about adaptive immunity. Second thing I want to talk about is all the parts of the adaptive immune system. You can see I also have 5 parts, A, B, C, D, and E. Just want to introduce each of these characters. First is your lymphatic system. This is really the battleground. This is basically a secondary circulatory system that you have in your body. So your primary circulatory system, of course, is your blood, and we've got a big pump called the heart. This is secondary to that in that there are vessels in the body, but there's no big pump, the heart. Instead, there's small muscles. So this system flows fluid, lymphatic fluid, and the lymphatic fluid flows a lot slower, mostly by tiny little muscle contractions. And it moves around lymph, which is full of white blood cells. The second thing are the antigens. So antigens, you're going to see some of these slides they're abbreviated as AG. So antigen, AG. This is the specific molecule found on the pathogens. So you can see I have some examples here. We have a bacteria and a virus. So on the bacteria, maybe we have a specific carbohydrate here that's found on the cell surface. On the virus, it looks like we have some sort of glycoprotein right here. So not the whole virus, but just a specific molecule on the virus. So you can see these are usually proteins, lipids, or glycolipids, or glycoproteins, of course, as well. Here's another example of a specific molecule, the diphtheria toxin. So the diphtheria toxin vaccine, for example, is just part of this protein. And so it's not even the full molecule, it's just part of the molecule that the immune system recognizes. The next character are these things called antibodies. So antibodies, notice I might abbreviate them as AB. They're also called immunoglobulins. So immunoglobulin, that really just means globulin is a type of protein found, and then immuno means part of the immune system. These are also abbreviated as IG. So both of these mean antibodies or immunoglobulins. So this is what an antibody looks like. It's actually 4 proteins. You can see that there's two longer strands. You can see they're called the heavy strand. There's one right there, a second heavy strand there, and there's these light strands here, 1 and 2, and they're attached together in this Y-shaped molecule. A couple of parts I want to point out. One is up here. This is called the variable region. This is the part of the antibody that recognizes the antigen. So if you're exposed to, for example, the measles virus or the measles vaccine, you're going to make antibodies that recognize something specific to that particular virus. This is what the antibodies are going to recognize and where they're going to recognize it. There's another region down here. This is the constant region. This part doesn't change. It's recognized by your immune cells. And so it's like an adapter between the part that attaches to the pathogen and the part that attaches to your immune cells. So here's a picture of some antibodies. You can see they're binding and recognizing different parts of this bacterial cell. You can see, for example, some of them are recognizing something on the surface here. Some of them are recognizing these little strand-like things. They're probably pili, so they're recognizing proteins or carbohydrates. It kind of depends on what that's supposed to represent. There's kind of a zoom in. You can see they can recognize different parts of these proteins, and each of these would be a different antibody. So you can see antibody 1, 2, and so on. So what do antibodies do? So 5 things really. We're going to talk about all of these things. We're mostly going to skip number 3 since we skipped the complement system in the last lecture. So we're going to talk about 1, 2, 4, and 5. Let's take a look at these. First one is opsonization. So what is opsonization? It really means just stimulating phagocytosis. I don't know why we need a special name for this, but we do have it. So you can see here is our little bacterium. It is bound by an antibody, and that is like a label that says to the immune cell, hey, come and take this and destroy it. Come destroy it by phagocytosis. So there we go. We've got a phagocyte. It's grabbing onto the bacteria. It's going to bring it in by phagocytosis, and it's going to destroy that bacterium. Number 2, antibody-dependent cellular cytotoxicity. So I called this before non-phagocytotic killing. We also call this cellular toxicity, so there's a few different names for this kind of thing. This, as I mentioned before, is kind of like the kiss of death. And you can see NK means natural killer lymphocyte. And over here, we've got a perforin. That's a toxin. And you can see that this toxin here is busting a hole in the side of the bacteria. So the kiss of death, it binds the pathogen, injects the toxin, and the toxin kills the pathogen. So whether you got phagocytosis or cellular toxicity, cytotoxicity, you're killing the pathogen one way or another. So what else can antibodies do? Sometimes they can agglutinate things. So agglutination really just means clumping. So if you look at these antibodies, they're Y-shaped and they have 2 binding surfaces. And you can see as a consequence, this can cause clumping of these organisms, whether this is bacteria or virus or otherwise. So what does this do? It basically makes a big clump, and the big clump is more visible to the immune system. You can see it says here this increases our chances of phagocytosis and reduces solubility and reduces the effectiveness of the bacteria to cause any sort of pathogenesis. You may have had your blood tested, whether you're A type, B type, AB type, or O type, and this is done through a process called hemagglutination, and this uses basically antibodies. So take a look at this. We're going to take a look right here. If you're B type, what that means. So if you're B type, you have the B antibodies, the B carbohydrates on your blood cells. And if you take a look at this, so what they're trying to show you is that if you're B type, so B type blood is right here, and you end up with clumping. Okay, if you have used the A antibodies right here, there's no clumping. If you use both, this is A and B, there's obviously clumping because you're going to get clumping every time you get B antibodies. Take a look here, here's A blood type, no clumping with the B, so no clumping here. We do get clumping with the A antibodies, we get clumping with both A and B antibodies. If you're AB type, you get clumping with B antibodies, you get clumping with A antibodies. If you're O-type, no clumping at all because you don't have the surface carbohydrates. So there's a practical use of agglutination. Okay, so the last thing that an antibody can do is basically neutralize something. So if you take a look here, this is actually a great case of something being neutralized. You can see this toxin here. This could be the diphtheria toxin. It is entirely neutralized by the antibodies, and so it can't go and cause any cellular damage. This is how the toxoid vaccines work, because they allow your body to neutralize toxins. You can see these bacteria over here and the virus over here. They're not able to invade tissues because they're neutralized. So just another thing that antibodies can do. So there are many types of antibodies, by the way. You can see they're labeled here, and you'll probably learn about some of these in different classes, and they are involved in different parts and different times of the immune response. And they all have their perks and their pros and cons. IgG, you can see here, it says it is important in all these processes. So there's opsonization, agglutination, we talked about those. The key thing about this one is it can cross the placenta. So a mother can give her fetus some protection from various diseases by sending these antibodies through the placenta, through the blood. So that one is important if you are a fetus, for sure. You can see IgA is the second one here. It can exist as a dimer, so it's kind of like two antibodies attached back to back. You can see this one is also important for babies because it can go in the mother's milk. So babies may not have a developed immune system, but their mother can help them out at least during the first few months of life and as a fetus. So IgM, this is a good one because it has all these antibodies attached together, 5 together. Very, very good molecule for finding and recognizing pathogens. This does all the usual things. You can see it's activating the complement, neutralizing, agglutinating, and so on. IgE, so this one here is very important for attacking parasites, and it turns out it's also involved in allergic reactions. And IgD, turns out we don't know what that one does yet. I'm sure somebody will figure it out someday. So what do we use antibodies for? We talked about these already as antibodies being used in all sorts of diagnostic techniques. We of course talked about the ELISA techniques. ELISA stands for enzyme linked, and so the enzyme is producing color. Over here you can say it's a yellow color, and usually these are done in some sort of multi-well plates, and these can be read by eye or by computer. There are these rapid test ELISAs we talked about. I showed you this slide before. This is a rapid HIV test. It's very similar to a pregnancy test. You can see there's two bands. There's a control band. If the control band should always work. If the control band does not work, as you can see in this case here, these are invalid because the control band did not work. Control band should always work. 2 bands mean positive, 1 band means negative. You can go back to your previous notes to look at these in more detail if you like. Okay, so the last part here of the immune system to talk about for the adaptive immune system before we talk about the processes, of course, are the cells. We talked about these different types of immune cells. Today we want to be talking about these lymphocytes found in the lymph. These are the T cells and the B cells. Now here's a little picture of them. You can see some neutrophils, some macrophages, and then the lymphocytes right here. So the B cells, B is for bone marrow. Bone marrow for B. You can also think of these found in the blood as well. So there's a B cell. The T cells, or the T lymphocytes, these cells mature in the thymus. So we have B and T. And there is an antibody. So what's going on with these antibodies? These are actually found attached to the cells. They're kind of like receptors. And they are antibodies. And so they are going to be interacting with the cell and interacting with the antigen. So a little bit more on those right away. So let's talk about these B cells and these T cells. What's going on in the bone marrow and what's going on in the thymus? I'd mentioned they mature there. This is a process called clonal deletion. So as I mentioned before, it's really important that our immune system does not attack our own body. So we do this by maturing these cells in the bone marrow and the thymus. So we eliminate all the cells that may potentially be reactive against your body. So T cells do this in the thymus, B cells do this in the bone marrow. Here's a little picture. You can see what's going on here is you have this here is a stem cell, and it makes a whole bunch of these immune cells. And in the bone marrow or the thymus, we're looking and we're realizing that, okay, oh, this one is going to react with ourself. This one too. We'll get rid of this one. We'll get rid of this one, and we'll keep these ones. So these are mature or trained lymphocytes that are ready for immune reactions. The other ones have been deleted or eliminated to remove potentially self-reactive cells. Another thing that these B cells and T cells do is they get selected. This is called clonal selection, and this happens in the lymph tissue, right? And so this is, as we mentioned, the battleground for the immune system is the lymphatic system. So how does this work? So there are those cells that we selected before. You can see it says they are naive. That means they haven't been activated yet. This one here recognizes an antigen. So maybe now you're exposed to the measles vaccine. And boom, it clones itself. This is clonal selection. Now we have cells that are active and ready to go, and some of them are going to be memory cells. So we'll talk about that process shortly. So there are many types of lymphocytes, particularly these T cells. I don't want you to get caught up too much in all the different types of T cells. There are more than 4 types. You can see some of them are involved in cytotoxic killing, some are involved in regulation, and all those type of things. Again, I don't want you to get bogged down in all the details. So one more thing is Part E, chemical signals and mediators, cytokines and interleukin and things like that. We are going to skip that this semester. Okay, so let's talk about the adaptive immune response. Okay, so there's 2 immune responses that we're going to talk about. The humoral response and the cell-mediated response. We're going to start off and talk about the humoral response. So the humoral response involves the B cells. And I always remember this because humoral, humor is an old word meaning blood. So B for bone marrow, or you can think of B for blood. So in this case here, the humoral response is going to involve these B cells. And these B cells, you can see here, they secrete antibodies, and the antibodies are going to do their thing. So we'll talk about that in a moment. First, what I want you to do is go and check out this video. I will have it attached to the course playlist with this particular lecture. So you can either type this link in here, or you can download the PowerPoint, click on the link, or you can go to the course playlist. So go do that right now. This is an excellent video that talks about what happens during the humoral immune response. It's a very good video. I think it was made in New Zealand. The guy has a cool accent, and please go check it out now. We're also going to talk about the cell-mediated response. I will come back to this in a minute, but this involves the T cells. So more on this in a minute. So the humoral response, let's just talk about that. You saw in the video there was a person getting strep throat, and you saw quite a few things happening. Let's kind of talk about what that typical humoral response looks like. So in this case here, you can see my step number 1 is there's some sort of antigen present. In the video, the antigen was present, I believe it was an M protein. Sometimes the antigens are actually discovered by those dendritic cells. We talked about those in the last lecture. They're kind of like little security guards that go around, and sometimes they find little pieces of things and they hold on to them for other immune cells to take a look at them. So basically the first step is the antigen is present, and it's either held onto by a dendritic cell, or sometimes they're just free, the antigens. Second thing that happens is a B cell, a naive B cell, you can see here it says here it attaches and it recognizes the antigen. And so once it recognizes the antigen, it gets activated. And what happens when it gets activated? First thing, it clones itself. Some of them become active cells, some of these clones, and these active cells secrete antibodies. Some of these cells become memory cells, so these are long-lived cells. We'll talk about those as well shortly. So let's talk about what happens with these antibodies. It's all the usual things. We talked about them just a few minutes ago. What do antibodies do? They neutralize things, so there's our toxins being neutralized or a virus is being neutralized. This can lead to opsonization. This is phagocytosis, like I said, just a fancy name for inducing phagocytosis. They can agglutinate, so form clumps, which usually leads to phagocytosis. Or lastly, they can induce some sort of other type of killing. So this is cytotoxic killing. You can see the toxins are being released by the natural killer. Okay, so we were just talking about the humoral response, and this involves B cells secreting antibodies. So the second adaptive immune response is called the cell-mediated response. This involves our T cells, and our T cells do not secrete antibodies. They target usually larger pathogens or other things that the immune system is going to have to deal with. You can see there are three things. Number one are infected cells. So what are we talking about? Usually cells infected by viruses. So viruses can infect your cells, and as they go through the replication process, they can produce things like glycoproteins that end up on the surface of infected cells, and these are not normal. They're not part of the human body, so they can be recognized by the immune system. Number two, the cell-mediated immune response can recognize cancerous cells. This is huge. This is very, very important for the immune system to recognize and destroy cancer cells. Cancer cells sometimes look a little bit abnormal. They don't look like normal cells, so they can be recognized by the immune system. Number three are pathogens that are too large to be destroyed by phagocytosis. So large parasites. So we're talking about parasitic worms and other things like that. So let's take a look at, I want you to go take a look at this video here. This is not found on YouTube, but I was able to find a very excellent video on the cell-mediated immune response. I encourage you to go check it out. It's a little bit longer than the last one, a little bit more detailed, but it's very well produced. So I encourage you to go check it out right now. So this finishes part A of this particular lecture. I wanted to try to keep these lectures to about 20 minutes max, so I'm going to break this up into two videos. See you in part B.